Thank you, Tiffany. Kia ora koutou. Ko hiu tako ingoa. He ka ako matua aho ki manaki whenua. Morning, everybody, and welcome to day 2 of our webinar series and our second weedy webinar. And we have Ronny Groenteman talking to us this morning. I always used to think of Ronny as our sort of junior scientist in our team, but Ronny's been with us for quite a long time now and has actually become one of our really senior scientists and now has a major role within our team and has recently begun a major project with MPI on control and research into weeds in New Zealand. So it's really an amazing transformation as time's gone by, and Ronny is now sort of one of the leaders of our teams. Aside from all of that, metabolomics, a rather new science and something which I still haven't really got my head around, but Ronny's going to tell us what it has to do with weed biocontrol. So take it away, Ronny. Thank you.
Tēnā koutou. Thank you everyone for joining me this morning. The work I'm presenting here today is actually the work of Paul Barrett, from Massey University, and funded partly by our Strategic Science Investment Fund through our Beating Weeds program. And Paul really is the expert on metabolomics. I'm certainly not an expert, but you know, you don't really need to know the ins and outs of a smartphone, for example, to be able to use it in a variety of ways. And similarly, all you need to know is enough about metabolomics to be able to think about how to use it for improving weed biocontrol. So my hope for today is that you take maybe 1 or 2 ideas from the ideas that I'll present here about how we can improve weed biocontrol with metabolomics, and maybe even spark your imagination to think about other applications that we haven't thought about. So let's get into it. The issue that probably sparked our interest in using metabolomics in weed biocontrol was that sometimes agents that we release fail to establish, or they might establish and fail to have an impact or have very little impact on the weed that they're released against. And when something like that happens, we would look at the issue from a variety of angles to try to figure out why. And this is one of the areas, one of the angles where metabolomics can fit in. So I'll give you an example to make it a bit closer to home. And I'll use the heather beetle as an example. And so the heather beetle was introduced in the 1990s and took over a decade, maybe closer to 2 decades, to properly establish and start having a serious impact on heather in the central North Island. And when that failure kept going, we tried to explore it from a variety of angles, starting with thinking about the intrinsic effects that could have affected the beetle's establishment. So the genetic bottlenecking that it experienced during the period in quarantine, in containment. That genetic bottlenecking probably contributed to a small body size. The small body size probably interacted with extrinsic effects, such as the long winters in the North Island Central Plateau, and potentially the extreme spring events, the ups and downs of temperatures. Other components, other angles that we might explore would be top-down effects, such as predators, parasitoids, and pathogens, which could be influencing establishment. And bottom-up effects. In the case of the heather beetle, this was something like the nutrition, so low foliar nitrogen. And then another element that can be added is perhaps the metabolites, the defensive metabolites profile of heather may be something that could have affected the beetle establishment. That is a question that metabolomics can help us answer. So a little bit about metabolomics. Where does it fit in the landscape of all of the other omics? We're all probably familiar with genomics, where we're comparing the whole genome of an organism to another organism. We can look at transcriptomics, where we're no longer looking at the whole genome, but looking at individual genes and their expression, whether their expression is elevated or reduced in response to a variety of stimuli. That expression, of course, comes into effect as proteins. So proteomics looks at the abundance of proteins that are the result of the gene expressions in response to stimuli. And then finally, those proteins, those enzymes are responsible for creating a variety of metabolites. And here comes metabolomics, which looks at the abundance of the variety of the wholesome of the metabolites in a sample, be it a sample from a cell or a specific tissue or an entire organism. In our case, we're looking at plants, so plant tissues. So the metabolome is the entirety, all of the identified metabolites from that particular plant tissue in our case, and that metabolome is the end result of all of those underlying processes that came before that, without us having to understand and know exactly what the processes were that led to that end result. Roughly, very roughly, how does it get done? We take a plant tissue sample, mix it with a variety of solubles, and that liquid goes into a liquid chromatograph. This is the device that Paul uses at Massey University. Broadly speaking, out of the chromatograph comes a chromatogram. And what I'd like you to take out of this sample chromatogram is the comparison here is between, say, a couple of samples, two samples. The black line is one sample, the red line another sample, and we don't know what that sample contains, what metabolites it contains. We just know that they're in a mass spectrum of around this size, and we know that the intensity of that metabolite is smaller in the red sample than it is in the black sample. So there's a difference in intensity of this metabolite. This is roughly the kind of results, outcomes that we get at that stage, without needing to know exactly what those metabolites are and what caused the differences. So Paul conducted a literature review around how well do we use metabolomics in wheat biocontrol, and the spoiler alert, we're not tapping that much well into that potential. So a little bit more detail about what the review finds. It finds that when we assess candidate agents for their safety and effectiveness as potential biocontrol agents, we seldom consider the biochemistry of the host plant. It also found that the plant biochemical phenotype determines the nutritional value and the chemical defenses in the plant. And so how well a herbivore, a natural enemy, can do on these plants. And that environmental conditions affect plant metabolomes. Now, I want to dwell a little bit on this point because there can be a couple of key mechanisms by which we get to that situation where the environmental conditions affect a plant metabolome. So one of them is a pre-invasion. We are talking about invasive plants. And if the plant has a pre-adaptation that really is a combination of traits that manifest as phenotypic plasticity that make the plant survive or even thrive in the new range, the new environment. The other mechanism is a post-invasion, and that could be through rapid evolution. And basically, that is where the environmental stressors in the new range select for suitable genotypes, and that ultimately manifests in a phenotype, in a biochemical phenotype that is different compared to the native range. But regardless of the mechanism involved, we end up in plants in a new environment, an environment that causes those plants and their phenotypes to adapt to the novel condition in a highly complex way that involves morphological, physiological, and biochemical changes. And we might end up with a situation where maybe on the surface the plants look the same in the native range and in the invaded range, but under the surface there are differences in the abundance of certain metabolites in those two ranges. And our question is, how does this affect herbivore performance? So these are abiotic influences on metabolome, and we're also then in the review looking at biotic influences. And so here, this is where the herbivory or effect of a pathogen, or if we're talking about herbivory, it could be different guilds of herbivores, such as leaf chewers or leaf miners or stem miners or piercing or sucking herbivores, how each of those guilds might affect the defense response, the biochemical response of the plant in a different way. So again, we don't need to know exactly the pathway and how it comes to effect, but we want to know the end result, the metabolic end result. So now I want to move on to 3 areas of application where we might be able to use this kind of technology in weed biocontrol. The first one is coming back to that issue of improving establishment that we talked about earlier. And this is around matching the target plant metabolomes at the collection site in the native range and the release sites here in New Zealand. So we know that small changes in the ratios of defensive plant compounds can alter the herbivore's attraction to the host plant or the level of performance on that host plant. But finding out exactly pinpointing which compounds and under which environmental conditions are responsible for those changes in performance, that is really challenging and costly. But if we look at the metabolome, at the end result, we're actually looking at a direct signature of the result of all of those biochemical processes. And it already encompasses, so those resultant compounds already encompass an imprint of all of the interactions between genetic, epigenetic, and environmental factors. So how do we bring it down to earth and how we might be able to use that in biocontrol? Again, let's go back to the example of heather. And Paul analyzed samples of heather from the UK and from New Zealand, and what we're looking at here on this graph is dots, where each dot represents the whole metabolome of one sample. And it's the pink samples come from New Zealand, the turquoise samples come from the UK, from the native range, from Scotland more specifically. And what we see here is that if we just
Hello, Ronny. Sorry, I just need to interrupt you. We've lost you temporarily there. I just need to let everybody else know we have had some internet issues with Ronny this morning and her presentation, and I just think she has almost disappeared on us completely at the moment. I'm not sure whether she can hear me or not. Oh, looks like she might be back again, so no.
this side, we just need to look for sites that show some similarity. And ideally, we would be looking for sites that sit closer as possible to that center of the grid, and that would be most resembling in their metabolomes. Again, without needing to know exactly what are the environmental factors that contribute to that metabolome. Another area of application is around effectiveness of agents. So selecting effective agents and trying to predict which agents would be effective prior to release is really key to getting us to the next step in biocontrol. It's very difficult for us still to predict which agents will be effective once they're released in the new environment. What we can do with metabolomics, there's two areas here, and one of them, the first one, is around looking at how much of a metabolic response, biotic response, biotic response, an agent or a candidate agent or a guild of agents elicit in the host plant. And certainly if the metabolic response is small and it doesn't look like those agents produce much stress in the plant, that could be an indication that it's perhaps not the greatest agent to pursue. On the flip side, if the response is high, there is a lot of stress in the plant and a lot of defense mechanisms start to come up. This is by no means a guarantee that this agent will be effective in the field, but it is perhaps an indication that it is more likely to be effective. And then another side to the effectiveness is perhaps an understanding when potentially an agent needs, a candidate agent needs a prior attack by another guild or another type of natural enemy to become effective, to elicit a strong stress response in the plant. So, for example, if a herbivore seems to be eliciting a low response, but perhaps it requires a pathogen to attack the plant first in order to allow the herbivore to create the stronger stress in the plant. Knowing that kind of information will tell us that we have to consider those kind of guilds, those kind of agents in a combination, and there is no point considering one of them without the other. The last area of application, again, has two components, two sides to the coin that we can think about using, and that's around augmenting our host range testing lists. And here in the picture, you can see Paul Barrett and Paul Peterson, the two Pauls, working on heather beetles. So the first side here is around adding to our test list species that are more distant phylogenetically from our target plant, but share a similar metabolite profile. So if you think about how we include plants in our host range tests list, we use phylogenetic relatedness. So we would start with plants that are in the same genus as the target plant, and then we would move further away to more genera in the same family, other families within the same order, and subclass, and so forth, getting further and further away. We call this the phylogenetic centrifuge, where we have the target species at the center of the centrifuge, and then distant circles, growing distant circles of plants in the same genus, other species in the same genus, more genera in the same family, and so forth. And so what we're saying here is there might be plant species that are really not related to the target species, but share a similar metabolic profile, and they could be added to the host range test list. So it's not going to replace the phylogenetic centrifuge, but it's going to augment it. That's a possibility. Another area, the flip side of that coin, is where we have plants that are very closely related to the target plant, but are potentially rare or protected, and we cannot obtain them for testing. And so if we could collect the metabolome of these plants and expose the agent, the candidate agent, to the metabolome, and hopefully the agent rejects that, that adds another layer of safety, another evidence for safety of that agent that just helps our understanding of our sense of security around our results with other closely related species. So finally, okay, how do we take this forward? what can we do with this now? One of the starting points for us can be a retrospective analysis of existing agents that are already well established in New Zealand. Some of them are effective, some are ineffective. And if we can compare what they do to the metabolome of their host plant, do they elicit a response, a strong response or not, we can start getting a little bit more stronger evidence around whether we can actually predict effectiveness by looking at the level of response, of metabolic response that is elicited by the herbivory. Another area is going back to the abiotic influences, and that is where we can start matching target plant metabolomes at the site of collection and site of release, and look at whether we get better establishment in sites that are metabolomically matched compared to sites that are unmatched. The technology is still a little bit too expensive for us at this time to start using it more routinely in the various stages of agent selection and host range tests. We may have to wait for these costs to come down, and we expect that they will. Same happened with molecular techniques. But in the meantime, until that happens, I do hope that you could get from this talk a little bit of an idea of the potential and how multifaceted it is. And that's all for me. Open to questions now. Thank you.
Great stuff. Thank you very much, Ronny. That's quite a interesting and scientific presentation on metabolomics, I have to say. So I'm hopeful that many people manage to follow that and understand more about the science and how we can utilize it in terms of our biocontrol future for host testing and establishment, and then following impacts and things like that. I assume that metabolomics is likely to have a role in measuring impact of our biocontrol agents on weeds in the future, Ronny?
Can you hear me?
Yes. Oops, you've gone again. You've come and gone on us.
Yes. The impact, I think we would still want to see more at the population level. Steps. Am I getting cut?
Yeah, you're coming and going, but that's right. Carry on.
Can you hear me, or am I getting cut?
You come and go a bit,
I
Ronny,
think
but
it
you're
could
okay.
potentially help us understand. Yeah, so I think it could potentially help us understand a little bit more of the details of maybe where we see impacts and where we don't. Maybe especially for agents that have an impact in some environments and not others. That could potentially help us look for more avenues to understand why, and maybe match better agents to those environments where we don't see impact. So potentially, yes, that's definitely another area to explore.
Cool. Okay. So there's a question here from Tammy Price. It says, how would you match target plant metabolome with collection site metabolome? Change the soil structure? Question mark.
So we're not going to do any action to match the metabolomes. Again, what we're looking here is we don't know what the processes are that cause that end result of a certain metabolome. What we would be looking is, we would be looking for sites. We would analyze plants at different sites in the native range and in the invaded range in New Zealand, and we would look for areas that match. So if I just go back to this graph here, here what we can see is that all of the sites, all of the samples have different metabolomes. Again, we don't know what caused that difference. Is it soil? Is it UV? Is it any other abiotic factor? We don't need to know that. all we need to know is that when we analyze metabolomes, if we can find samples that fall close to each other, and hopefully close to the center of the grid, samples from the native range and from New Zealand that fall close together, then we know we've got sites with matched metabolomes. And that's all we need to know. We don't need to know how they came to have that similar metabolome. We're just interested in that end result. And that is part of the beauty of the method, that we don't need to understand the ins and outs of how it got to that point.
Cool. Okay, thank you. Another question here from Andrea McCormick. Which weed species would be interesting for further metabolomics analysis?
I think, again, as I mentioned at the potential first steps for us, it would probably be everything at the moment in New Zealand that has matured biocontrol programs, where we can say that certain agents have been effective or have been ineffective. So starting to gather that baseline information, that baseline of evidence for whether metabolomics can be a good predictor for future programs based on that evidence from past programs. I think that would be the most interesting part for us now, building up that evidence. Yeah.
Cool. Excellent. Thank you. Hester Williams has asked, is it useful in cases where secondary metabolites are not identified or well known?
I think you got cut a little bit, so could you please repeat the question?
Sorry. Okay. Hester says, is it useful in cases where secondary metabolites are not identified or well known? Oh, we lose again,
Yes,
Ronny.
yes, it is. Okay, can you hear me now?
Yep.
I think yes, it is, again, because the point is that we don't need to know the exact details of which metabolites and where they're going up or down. At least for some of those applications, all we need to know is the similarity or the dissimilarity between the metabolomes here, for example, at the different sites. Where we talk about the biotic examples, about the impacts of herbivory or pathogens on the plants. It's enough to know that perhaps those metabolites are from groups associated with defense mechanisms. We don't need to know the exact metabolites.
Okay. That's great, thank you. Frank Millennia says, great talk, Ronny, but any idea how quickly the metabolic response occurs in plants? This might impact when to measure for these things.
Now you're getting into the sides of the ins and outs that I'm not qualified to answer about. Look, things can happen pretty quickly. I would definitely tell you that when you collect the sample, you've got to kind of fix it very quickly, or there will be obviously changes in metabolites as soon as you cut the leaf, probably, because you are putting it into stress. With abiotic conditions, maybe it's a bit slower, but I guess you could compare in comparable seasons, probably. But I think I would need maybe one of the better experts on the technologies to actually answer that conversation.
Okay, thanks, Ronny. Another question from Shane McNeil. Is woolly nightshade a potential candidate species? The lacebug appears to have mixed rates of success across its range.
Potentially, yep. Yeah, it is certainly one that was on my mind when I answered one of those previous questions. But certainly that's an agent where a weed where we see a certain agent affecting it more in some environments, so shady versus sunny environments, better control in one rather than the other, and yeah, whether metabolomics has anything to do to give us a clue on how to improve control for those other situations. Absolutely.
Yes, I think we've actually got plenty of examples of that sort of thing in the history of our weed biocontrol programs, of where things have worked in one place and not in another, or have had impacts in one area and not in another, and why those things are true. Cool. Reese Burns has a question here. Would you look at metabolomes in roots for agents that attack roots, leaf metabolomes for agents that attack leaves, etc.?
My logic potentially says yes, but again, I think we would have to ask more of the experts. It sounds sensible to me.
So, and Andrea's made a couple of comments here, but she says that we collect lots of environmental data and conduct experiments under controlled conditions to explore individual factors. So Andrea may be able to provide some more information about one or two of those questions as well. So you'll be able to talk to her and answer some of them afterwards if you need to, Ronny. All right. That looks about it. Well, thank you very much, Ronny. Really cool. We got a few technical glitches in there, but we overcame them. So it worked out really well. Thank you very much for your presentation, and thank you to everybody else for your attendance. And we'll see some of you perhaps tomorrow for the next one. Otherwise, thank you very much, guys. See you later.